Good day, guys. Sir Levy is back. This is Psycho 101. So last time we talked about the schools of psychological thought and the branches of psychology. Let's now move on to one of the branches of psychology, which is biopsychology. So at the end of this video, the student is expected to 1. Define the emerging branch biopsychology. Second, the student will be able to discuss the structural units and bioelectrochemical processes in the body form as basis of the nervous system. And third, you will be able to point out the divisions and integrated functions of the connectors, effectors, and the receptors as the foundation of behavior. You will be also expected to explain the effectors or muscular and glandular systems and their effects on behavior. And lastly, you will be able to locate and discuss the major parts and functions of receptors or sensory organs to the behavior. So, let's get going. So, ano ba yung tinatawag na biopsychology? So, Zimbardo, Weber, and Johnson in 2000 defined biopsychology as the specialty in psychology that studies the interaction of biology, behavior, and the environment. So, Fieldman also refers biopsychology as neuropsychology, with which the importance of the nervous system in controlling behavior has given major emphasis on the biological underpinnings of behavior. So, according to Zimbardo, Weber, and Johnson, ang biopsychology is the interaction between and among biology, behavior, and the environment. So, biology talks about human being as a living creature, and of course, there is behavior whenever they are exposed to a certain environment. So, this kind of interaction is known as biopsychology, according to Zimbardo, Weber, and Johnson. According to Feldman in 2002, sinasabi niya na ito din ay maitatawag na neuropsychology, which deals with the study of the importance of the nervous system o yung ating brain in controlling behavior. So according to this biopsychology, behavior is based on the reactions inside the brain or the nervous system. So biopsychologists and neuropsychologists seek to understand the neurological basis and biochemical process of behavior. So ano yung basihan ng behavior ng tao base sa kanyang brain and base sa mga chemicals inside his body. So by understanding the important divisions, parts, and functions of the nervous system, this can explain the bodily processes and behavior of the organism. So yung mga biopsychologists natin at neuropsychologists natin ay pinag-aaralan yung mga reactions or stimulus and the reactions of the brain. Ano ang nagtutulak na chemical sa ating brain upang ito ay mag-react or magbigay ng signal sa iba't ibang parte ng katawan to react. So mga specialists natin ay concerned with the basis of voluntary and involuntary functioning of the body. So for example, yung ating mata, bakit tayo pumipikit? And that is an involuntary function of the body. Kusa na lang natin itong ginagawa without our permission. And there are times we are voluntarily acting based on our will. Okay? So their scientific study shows that the transmission of neural impulses is conveyed to and from the central nervous system mula sa ating central nervous system at papunta sa ating buong katawan ay may transmission ng neural impulses o yung tinatawag ng ilan neuroelectrical impulses o para siyang electricity na nagsisend ng message from the brain sa iba't ibang katawan and from iba't ibang parte ng katawan to the brain. The specialist examines the location and structure of the brain and its effect on behavior. They can discover the causes of psychological disorders to be traced to biological factors so that appropriate treatment can be effectively applied. So through this study of biopsychology, malalaman natin kung paano natin i-deal o aayusin ang certain behavior. How are we going to interfere with the pattern of behavior at baguhin ito? So one of the major reasons why we are studying psychology is to control behavior. So how can we control behavior if we do not understand the cause of it? So ang sinasabi ng biopsychology ay nagmumula ito sa brain at sa mga chemicals that are being released by the brain. So after ma-identify yung cause, of course, we can apply treatment to it. Madali nang baguhin o kontrolin ang behavior. Of course, bear in mind that understanding of human behavior cannot be complete without knowledge of fundamentals of the brain and the rest of the nervous system. So we need to identify kung ano ba yung pinagmulan ng behavior, at of course, hindi natin maiintindihan yan kung wala tayong fundamental knowledge of the brain, and of course, not only the brain, but the whole nervous system, including spinal cord and other parts of it. Biological factors cannot be overemphasized without the strong impact on sensory experiences o yung mga nararanasan natin like nakikita, naamoy, naririnig, natitikman, nasasalat, states of consciousness, motivation and emotion. Siyempre, ano yung laman ng ating consciousness o ano yung estado ng ating consciousness o ating pag-iisip, anong nagmo-motivate sa atin, including our emotion o emosyon. Emerging branch of biopsychology had paved the way for the treatment of drugs and other treatments for psychological and physical disorders. In short, biological factors are placed first in the hierarchy of diagnosis, followed by psychosocial and sociocultural makeup. Una nang kinukonsider ngayon ang biological factors kung bakit nagkakaroon ng certain behavior, psychosocial or sociocultural behavior as a whole. So ito na, the basic structural unit of the nervous system called nerve cell or neuron, as illustrated in figure 2. Neurons are staggering an approximation of trillion neurons throughout the nervous systems, and they are involved in controlling the whole body and the behavior. So ang basic unit ng nervous system natin ay tinatawag na neuron, at yan ang nagtatransmit ng message from the brain to the whole parts of the body. So ito yung nagpo-control ng ating behavior at pati na rin ng ating actions. Yan nung ito ang structure of the neuron natin. As you can see, we have here dendrite, the nucleus, the axon, the cell body, the node operandi, myelin sheath, Schwann cells, and action terminals. Ito ay ang parang electric impulse na umiikot sa buong katawan natin na may bit-bit na message from dendrite to axon terminal to another neuron. So like other cells in the body, neurons consist of cell body containing a nucleus. So yung cell body natin, sa loob ng cell body ng neuron, we have here the nucleus, at itong nucleus ay naglalaman ng materials that provides nourishment and insulation. Likewise, they establish how the cell will function. Neurons have various functions. They communicate with other cells and transmit information. From cell to cell, nagtatransmit siya ng information while they are communicating with each other. So they have clusters of fibers called dendrites at one end. So from one end of the neuron, you can see some kind of roots na para siya mga ugat. So that's what we call dendrites. Dendrites appear to be the twisted branches of a tree, more particularly receive messages from other neurons. Ito ang nagre-receive ng message from other neuron because we have trillions of neurons inside our brain and the whole body. So at the opposite end, neurons have a long, slim, tube-like extension na tinatawag na axon. So the part of the neuron that carries messages to other neuron. The axon is relatively longer than the other neurons. Most axons have several millimeters in length, but some axons can be as long as 3 feet. So yung axon ang pinakamahabang parte ng ating neuron. That can be as long as 3 feet. Axons terminate in small bulges called terminal buttons that send messages to other neurons. Dito na siya nagtatapos sa terminal buttons. Imagine the trillions of neurons, the whole body circulating. Guys, ganun mag-send ng message ang ating nervous system to the different parts of the body. So what makes this neuron special and unique from different other kinds or types of cells? So ito yung mga characteristics natin na distinguish sa neuron. Number 1, specialization for conduction of impulses. So ito ang nagdadala ng impulses natin. So, great sensitivity to oxygen deprivation. Automatic kapag ito kinukulang ng hangin, napaka-sensitive dito. So, number 3, importance for many vital functions. So napakalaga nito in different vital functions, and the fact that they do not multiply, with which responsible for so many of the incurable conditions such as paralysis, chronic vegetative states, palsy, blindness, and etc. So hindi ito nagmo-multiply, so therefore mentras tumatanda tayo, nababawasan yung ating neurons. So now we have these three types of neuron, okay, as presented in the picture. So first we have the sensory or afferent or bipolar neurons, then motor or moto, efferent or multipolar neurons. Then 3 is the association, central adjusting interneurons or pseudopolare. So what is a sensory neuron? As you can see in the picture, the first neuron is the sensory neuron. So the work of the sensory neuron is to transmit impulses initiated by receptors to the spinal cord and the brain. So from the receptors or sensory receptors, ito yung mga receiving area natin gaya ng sense of touch, sense of sight, magmumula yan sa mata, sa skin, and other parts of the senses natin. Magsisend siya ng impulse going to the spinal cord and the brain. So receptors are specialized cells in the sense organs. Ito nga yung sa may muscles, skins, joints that detect physical or chemical changes and convert these events into impulses that pass along the sensory neurons. So kung ikaw ay magkakaroon ng cut sa skin, automatic marireceive siya ng tinatawag nating sensory receptor, and the impulse will be sent to the spinal cord and to the brain. At yung signal na yon ay magsasabi sa brain natin that there is pain. So that's what we call sensory neuron. Nagdadala siya ng mga impulses initiated by the receptors na nagmumula sa ating mga sense organs. Next, we have this motor or moto or efferent or multipolar neurons. So they carry impulses away from the brain. So kanina, from the different sense organs, papunta sa brain natin. This time, away from the from the brain, okay? So they carry impulses away from the brain and spinal cord to reacting effectors, organs, the muscles, and the glands. Magsisend na siya ng signal or impulses away from the brain going to the different parts of the body as the effectors or the organs, the muscles, and the glands, at yun ang magre-react. So sabi ko nga, if you felt pain, from the pain or from the skin, magdadala ng mensahe yan from the receptors going to the brain. Ang tawag natin is sensory neuron. At yung magdadala naman from the brain to the different parts of the body or the sense organs or the organs natin, muscles and glands natin, nagdadala naman ng neuron na yun ay yung motor neuron or moto neurons. Okay? So yun ngayon, kapag ka naramdaman mo na yung pain, automatic yung kamay mo kusang, yung reflex kusang kikilos yan para hawakan yung nasugatan o para maprotektahan mo pa yung nasugatan. So that's motor neuron. And the next, it is actually in the picture, it is in the second picture of the neuron. So next is association, central adjusting interneurons o yung pseudopolare. So they receive impulse from the sensory neurons and carry impulses to other interneurons or motor neurons in the brains, eyes, and spinal cord. Ito, neuron to neuron. So ang nagtra-transmit ng impulse from a neuron, it could be a sensory neuron or a motor neuron, to another neuron, okay, that's what we call interneuron, okay, or association, central adjusting interneurons o yung pseudopolare. So that's the third picture in the presentation. So the signals transmit via neuron are charged purely electrochemical by nature. So therefore, para siya electricity that flows over and circulating inside the body. The electrochemical charges or impulses generally move across neurons when they were traveling on a one-way street. So from neuron to neuron nagta-travel yung ating tinatawag na electrochemical impulse. They follow a route that initiates by the dendrites. So si dendrites mismo ang nag-initiate by transferring, okay, para sa daan, okay, continually into the cell body and brings ultimately down the tube-like extension na tinatawag na axon. Yan nga yung pinakamahaba nating parte ng neuron, no? Dendrites then detect impulses from other neurons, i-receive ni dendrite. Actions carry signals away from the cell body, mula doon sa cell body, nucleus, cell body, and dadaan doon sa axons, at doon lalabas sa terminal o doon sa pinaka neuron terminal natin, no? So then transfer to another dendrite, another neuron na naman. So yung myelin sheath naman natin ay nagse-serve as a covering sheath doon sa pinaka action natin. So myelin sheath is covered with a thin fatty tissues and protein cells that protect themselves around the action in preventing messages from short-circuiting one another. Baka kasi mamaya magdikit yung mga neurons natin, mag-short-circuit, para nga siyang kuryente. So because actions must be insulated in some way, just as electrical wires must be insulated as in the case of a car battery. So the myelin sheath serves to increase the velocity o yung bilis ito with which the electrical impulses transmit from the dendrites to the end of the action vary from 2 to 200 miles per hour. Imagine kung gaano kabilis mag-transfer yung ating impulses o yung message from other sensory or motor neurons natin through the sense organs, then passing through the brain into the different parts of the body. Grabe, kaya pala napakabilis ng reflex natin pag may tatamang something sa ating mata, magsesend na ng message sa ating eyes na kailangan na niyang pumikit. So napoprotektahan natin yung eyes natin. So that's how fast the impulse circulating around the body, no? So the diameter of the axons, that those larger ones generally being faster. The larger it is, the axon, mas mabilis ang pag-transfer ng impulse. And then the axons that carry the most fundamental information have the greatest concentrations of myelin, kasi pinoprotektahan nito ang axons nang sa ganun hindi magkaroon nga ng short circuit. So for example, the information regarding the pain is carried via axons. Dala-dala ni axon yung pain impulse natin in the hand and the arm that have a considerably thick coating of myelin. Then the sudden velocity of the message of pain to the brain. So kaya pag nagkasugat ka sa kamay o sa paa, automatic nararamdaman mo yung pain. That's because of the axons protected by the myelin. The sudden velocity of the message of pain to the brain in certain disorders like multiple sclerosis, a disease characterized by the severe sensory and motor nerve dysfunction, maaring ma-delay o kaya naman hindi mo maramdaman yung pain, is due to the degeneration of the myelin sheath. So meron nga tinatawag na degeneration of the myelin sheath o yung demyelination. So sheath that enveloping the axon to deteriorate. So therefore, more or less, ang reason kung bakit nagkakaroon ng sclerosis is degenerated na yung myelin sheath at hindi na napoprotektahan yung axon. So therefore, nagkakaroon ng short-circuiting. The exhibiting parts of the axon that are hidden would produce to short circuits messages between the brain and the muscles. Such results in symptoms would lead to the inability to walk, hindi ka makakalakad because of short circuiting, visual problem, pwede ka magkaroon color blindness, farsightedness, and general muscle dysfunction. So moreover, the nervous system consists of a huge number of non-neural cells called glial cells. Glia is a Greek word meaning glue that are scattered among neurons, with which the main function is to hold the neurons in place. So meron tayong tinatawag na non-neural cell o hindi siya neuron, hindi siya nagpa-pass ng message from one neuron to another neuron or passaging cell. Itong non-neural cell na to ay tinatawag na glial cells. Ginagawa lang siyang parang glue, okay, that are scattered among neurons, with which the main function is to hold the neurons in place. So in order for the neurons to stick with one place, yung glial cells po yung nagpoprotekta dyan at parang ginuglue siya sa isang place lamang, okay, so that it can protect the neurons. So guys, just remember the name, okay? Glial cells. It glues the neurons in one place, or holds the neurons in one place. Just bear in mind that glial cells are not responsible to receive or carry impulses, but rather they provide structural and metabolic support. Therefore, support lamang siya, and provide in other ways to ensure that neurons can perform their functions. Like a firearm, para siyang baril, neurons either shoot or do not shoot have no in-between state, just as pulling harder on a gun trigger does not make the bullet move faster or move more certainly. So therefore, parang baril siya kahit gaano kadiin pa ang pagpindot mo doon sa trigger or pulling the trigger, it doesn't mean it would make it faster. So ganun din ang glia cells. Parang support lamang siya pero hindi ito pinapabilis o kung ano pa man, but it just protects the neurons. Likewise, neurons follow an all or none law, with which neurons are either on or off with nothing in between the on or off state. Once triggered beyond a certain point, a neuron strikes. Once na-trigger ang isang neuron, automatically para siyang kuryenteng magpa-pass through to another neuron. In resting state, a neuron has a negative electrical charge of about 70 millivolts. A millivolt is 1,000 of a volt as shown in the right side. As you can see, ganyan ang itsura ng ating neuron kapag ito ay resting state. This electrical charge is caused by the presence of more negatively charged ions within the neuron than outside it. Inside of the neuron is representing the negative pole, and the outside of the neuron is numbering the positive pole. When an impulse starts to fire, the cell membrane in the neuron allow positively charged ions to rush in at rates as high as 100 million ions per second. The speed arrival of these positive ions causes the charge within that part of the cell to change momentarily from negative to positive. So when the charge reaches a critical level, the trigger is pulled and an electrical nerve impulse known as an action potential travels down the axon of the neuron. The action potential travels from one end of the axon to the other like a flame traveling across a fuse toward an explosion. As the impulse moves along the axon, the movement of ions causes a sequential change in charge from negative to positive. After the passage of the impulse, positive ions are pumped out of the axon, and the neuron charge returns to negative. Just after an action potential has passed, the neuron cannot fire again immediately, no matter how much stimulation it receives. For you to see what's really happening upon triggering the impulse of a neuron, let's watch a short video, okay?
This is a neuron, which has four main parts. The dendrites receive information. The cell body processes and integrates that information. The axon carries the information along long distances from one part of the neuron to another, and the axon terminal transmits the information to the next cell in the chain. A bundle of axons traveling together is called a nerve. Nerves can be very long, as they often need to transmit information over long distances. As we just saw, the dendrites are the part of the neuron that receives incoming signals. Based on the strength of this incoming stimulation, the neuron must decide whether to pass that signal along or not. If the stimulation is strong enough, the signal is transmitted along the entire length of the axon in a phenomenon called an action potential. When this happens, we say the neuron fires. Transmission of a neuronal signal is entirely dependent on the movement of ions or charged particles. Various ions, including sodium, potassium, and chloride, are unequally distributed between the inside and the outside of the cell. The presence and movement of these ions is not only important when a neuron fires, but also at rest. To start, let's think about the positively charged sodium and potassium ions. When a neuron is not sending a signal, it is considered to be at rest. In a typical neuron in its resting state, the concentration of sodium ions is higher outside the cell than inside. The relative concentration of potassium ions is the opposite, with more ions inside the cell than outside. This ionic separation occurs right at the cell membrane and creates a chemical gradient across the membrane. Because ions are charged particles, we also need to consider their charge when thinking about their distribution across the membrane. At rest, there are more positively charged ions outside the cell relative to the inside. This creates a difference in charge across the membrane, which is called an electrical gradient. Together with the chemical gradient we already mentioned, we refer to this ionic imbalance as the electrochemical gradient. The difference in total charge inside and outside of the cell is called the membrane potential. At rest, when no signals are being transmitted, a neuronal membrane has a resting potential of approximately -70 millivolts. This means that the inside of the cell is approximately 70 millivolts less positive than the outside. Both the chemical and electrical gradients we just discussed contribute to establishing this potential. While the inside of the cell has a net negative charge and the outside of the cell has a net positive charge, the charges line up at the membrane and the bulk solution on either side is actually electrically neutral. The resting membrane potential is the point where the cell has achieved electrochemical equilibrium. This means that the concentration gradient and the electrical gradient for each ion is equal and opposite. Ions cannot simply move across the membrane at will. Instead, they need a protein embedded in the membrane to facilitate their movement. Most ions cross the membrane through a structure called an ion channel. Ions move through channels by passive diffusion along their concentration gradient. Some ion channels are always open, but many require a signal to tell them to open or close. For example, voltage-gated channels only open when the membrane potential reaches a certain value. On the other hand, ligand-gated ion channels are triggered to open when they are bound by a specific molecule. Mechanically gated ion channels open in response to physical forces such as changes in length or changes in pressure. Most ion channels are selectively permeable, meaning that they only allow one or a small subset of ions to pass through. Voltage-gated ion channels, for example, typically only allow a single ion to cross the membrane when they open. This means that we need separate channels for each ion, i.e., voltage-gated sodium channels as well as voltage-gated potassium channels. As ions move through a channel and cross from one side of the cell membrane to the other, they cause the membrane potential of the cell to move away from its resting potential. If the resulting change in membrane potential is small, we call this a graded potential. Graded potentials can vary in size, can be either positive or negative, are transient, and typically do not result from the opening of voltage-gated ion channels. When ion channels open and a graded potential occurs, the neuron moves quickly to reset its membrane potential to resting values. This is accomplished primarily by the use of the sodium-potassium pump, which uses the energy generated by ATP hydrolysis to actively transport ions across the membrane against their concentration gradient. In other words, sodium is transported to the outside of the cell, where its concentration is higher, and potassium is transported back into the cell, where its concentration is higher. One cycle of this pump transports 3 sodium ions outside the cell and brings 2 potassium ions inside the cell. This unbalanced charge transfer contributes to the separation of charge across the membrane and also to the ionic concentrations we see at rest, thus restoring the chemical and electrical gradients to their resting levels. Maintaining these ionic balance in neurons is so important that this process can account for 20 to 40% of the brain's total energy use. Only when the resting membrane potential and ion distributions are maintained at precise levels will the neuron be poised and ready to fire an action potential. When the outside stimulation is large enough to bring the membrane potential in the neuron body up from -70 millivolts to the threshold voltage of -55 millivolts or higher, this triggers an action potential at the axon hillock, which then travels down the axon. Voltage-gated sodium channels have 3 states: open, closed, and inactivated. At rest, the sodium channel is closed. Once the cell membrane reaches the threshold voltage, the channel changes to an open position, and sodium rushes into the cell because of the electrochemical gradient. As positive sodium ions enter the cell, the membrane potential becomes less negative and more positive as it approaches 0 millivolts. This is called depolarization. Eventually, the voltage gradient goes to 0 and beyond 0, up to a positive 30 millivolts. This is called an overshoot. As the membrane potential becomes positive, the sodium channel inactivation gate shuts, making the channel inactivated. This stops the flow of sodium ions into the cell. The change in membrane potential also opens the voltage-gated potassium channels, though they open and close more slowly. Because of the potassium electrochemical gradient, potassium ions flow out of the cell, making it less positive and eventually negative. This process is called repolarization. Because the potassium channels are a little slow to close, for a brief period the membrane potential is hyperpolarized. It's more negative than the resting potential. During hyperpolarization, the potassium channels close. Throughout all this, the sodium-potassium pump is still working. The pump restores the chemical gradients by putting the sodium and potassium back in place. And the pump reestablishes the potential gradient by moving more sodium ions out than potassium ions in. This returns the membrane potential back to its resting potential. During repolarization, the inactivated sodium channels won't respond to any stimulus at all. During this time, the neuron is in its absolute refractory period, the period of time when a nerve cannot fire another action potential no matter how strongly it's stimulated. The absolute refractory period prevents action potentials from happening again too quickly and prevents the action potential from traveling backwards along the axon. During hyperpolarization, the sodium channels are closed and the inactivation gate opens. There is no change in sodium flow, but now they could be opened again. This is called the relative refractory period, because while the sodium channels could open, it would take a larger than usual stimulus to reach threshold, because the cell is hyperpolarized due to the potassium still leaving the cell. The amplitude of the action potential for a particular neuron, that is, the maximum voltage in one neuron during an action potential, never changes. An action potential doesn't get bigger with a bigger stimulus. It's all or nothing. It either happens or it doesn't happen. What can change is the frequency of the action potential. A neuron might fire many more times per second in response to, say, an intense pain, and less frequently in response to a gentle breeze. Some axons transmit action potentials faster than others. One variable that increases conduction velocity is the presence of myelin sheaths around axons. Myelin speeds up transmission through a process called saltatory conduction, in which the action potential signal appears to jump along the part of the axon covered by the sheath. In the peripheral nervous system, the sheaths are formed from glial cells known as Schwann cells. There are small gaps between Schwann cells called the nodes of Ranvier. The action potential appears to jump from node to node, speeding the transmission. In the central nervous system, the sheaths are made by cells known as oligodendrocytes. To review, with no stimulus, the membrane is at its resting potential. A small stimulus causes a graded potential, and a stimulus above the threshold creates an action potential and the neuron fires.
Okay, so that's everything about the neurons. Next time we will be discussing the different kinds of chemicals that could affect the neurons. For now, it's time to say goodbye, so see you next time, guys.